Hello everyone. Today our group will explore the cutting-edge intersection of biochemistry and environmental science. We're focusing on bioremediation, the vital process that uses biological agent to neutralize dangerous contaminants in our ecosystem. In 2025, global pollution remains a severe threat to our health and the planet's ecological balance. We see this in the dangerous accumulation of heavy metals, hydrocarbons, and synthetic plastics. While traditional cleanup methods exist, they are often too expensive and can even cause secondary damage to the environment. Because of this challenge, we turn to the molecular engines of nature, biological macromolecules. Bioremediation relies entirely on the biological capabilities of microorganisms and their fundamental components, DNA, RNA, and proteins. But how do we know if these microbes are actually doing their job? This is where omics technology come in. This toolkit allow us to move past simple trial and error and instead use a system biology approach to understand the genetic and enzymatic mechanism driving environmental cleanup. Next, we'll dive into a specific biological arsenal, the proteins and nucleic acids that make this cleanup possible.
Today, everyone, I am Prisa Dukinlay. Today, I will be discussing the biological arsenal, macromolecules, and action. Bioremediation is rooted in the central dogma of biology, where DNA encode pollutant-degrading enzymes. RNA regulates gene expression, ensuring microbes produce the necessary proteins to break down environmental contaminants. Moving on to enzymes, the molecular workhorses. Enzymes are the primary workhorses of bioremediation. Oxidoreductases degrade phenolic pollutants, while hydrolysis break down oil and pesticide molecules by lowering activation energy and enabling efficient chemical bond cleavage. Now discussing transport, proteins, and metal sequestration. Beyond enzymatic degradation, transport proteins and microbial membranes facilitate the uptake of heavy metals such as chromium 4. Inside the cell, enzymatic system reduce chromium 4 into the less toxic, which is chromium 3, helping detoxify contaminants and protect groundwater. Overall, biological macromolecules contribute to environmental remediation by enabling microorganisms to restore polluted ecosystems.
To understand how microbes clean polluted environment, scientists use a powerful strategy called the multi-omics approach. The genomics and the metagenomics reveals the genetic potential of site by identifying all the DNA present, including K degradation genes like ALK-B. The transcriptomics shows which genes are actively expressed, helping us identify microbes that are truly breaking down pollutants. Proteomics identifies the functional enzyme produced, confirming that the genetic instructions are translated into actions. The metabolomics analyzes the end products, ensuring pollutants are converted into harmless substances like carbon dioxide and water.
And now we were going to talk about NGS, or the next generation sequencing. NGS is like a high speed DNA reader that helps scientists understand the genetic information of organisms much faster and more accurately. It helps identify organisms and genes directly from samples without the need for laboratory culturing. NGS is also important in sequencing DNA directly from environmental samples. It allows scientists to study microbes in soil and water without growing them in laboratory. It also solves the great plate count anomaly. Since over 99% of microbes cannot be cultured, NGS reveals the true microbial diversity that traditional methods miss. Next generation sequencing also improves bioremediation strategies. By understanding microbial rules and interactions, scientists can design targeted, efficient, and eco friendly environmental cleanup methods. NGS also shows how microbial communities work together. Different microorbs perform different steps in breaking down pollutants, making cleanup more effective. And lastly, NGS identifies which microorbs are present in polluted environment. This helps researchers know which organisms have the potential to degrade specific pollutants.
To conclude our presentation, biochemistry is important in environmental science, especially in bioremediation. Biological macromolecules are the key components that break down harmful pollutants, making environmental cleanup possible. With the use of omics technologies, we can deeply understand and improve these biochemical processes. This shifts bioremediation from simple trial and error to a more precise and science-based approach, leading to cleaner and more sustainable solutions for our environment.